Assalamu alaikum students. Our next topic is the glycoprotein and its structure. Uh, you people know that sometimes carbohydrate binds with the protein and sometimes binds with the lipids. So the carbohydrate bonded with the proteins are called the glycoproteins and bonded with the lipids are called the glycolipids. Proteins that have covalently bonded carbohydrate units attached to them, such kind of proteins will be known as the glycoproteins. These proteins are involved in many biological functions, including they may produce the immunological protections. They may be involved in the cell-to-cell recognitions. They may be involved in blood clotting, cell transduction, signal transduction processes, and sometimes host and the pathogen interactions. So there are many biological functions of the glycoproteins. Carbohydrates portion of a glycoprotein usually varies from 1% to 30% of the total weight. Some glycoproteins may contain as much as 50% to 60% carbohydrates, but mostly they are usually composed on 1 to 30%. The most common monosaccharides found in the glycoproteins are the glucose, mannose, galactose, fucose, N-acetyl galactosamine, N-acetyl glucosamine, and N-acetyl uh, neuramic neuraminic acid. Yeah, we can call it the sialic acid. These are the structure of these compounds. These are the structure of the monosaccharides which can be attached with the glycoproteins. Sugars are attached to the proteins as branched oligosaccharide units, usually containing fewer than 15 carbohydrate residues. As far as the linkage is concerned, there are two important types of the covalent linkage which are involved in attachment with the sugars to the proteins. There could be there are two types of the there are two types of proteins can also be linked through the O-glycosidic linkage and through the N-glycosidic linkage. The example one is the O-glycosidic linkage, where the polypeptide chain containing the amino acid threonine is being attached with the N-acetyl galactosamine through the O-glycosidic linkage, and the lower polypeptide chain in which the asparagine is being attached with the N-acetyl glucosamine with the N-glycosidic linkage. Additional carbohydrates are usually linked to the first monosaccharides on the glycoproteins. There may be attachment of the chains. Here you can see that here asparagine is being attached with the through the N-glycosidic linkage to the N-acetyl glucosamine. This N-acetyl glucosamine through the beta-1,4 linkage is attached with the other units of the N-acetyl glucosamine, and then there are further mannose subunit which are making the branches uh, which which are making the branches of such which are making the branches for this linkage. These mannose residues can be attached through the alpha 1,3 and the alpha 1,6 linkage. As many as mannose residues will be attached, the branching pattern will be increased. Glycoprotein with the unique oligosaccharide compositions and the sequences are said to be the information rich. They are frequently bounded. They are frequently bounded on the cell surface, where they serve as the marker to identify the type of the cell. Recognition of the surface oligosaccharide is involved in diverse processes: blood type determination, antibody action, cancer initiation, turnover of the age proteins, and viral growth. It has also been discovered that attached sugar tail to medicinal proteins enhances the therapeutic potency of the proteins. Many new discoveries about the structure and the action of the glycoproteins are coming from the study of proteins that act as antifreeze agent in the blood of Arctic fish living in sub-zero waters. Function of the glycoproteins and the cancer. When cells transform from a normal state to a malignant state, changes takes place in the glycoprotein on the cell surface. These changes, usually caused in the cancerous cells, by a deficiency of enzyme called glycosyltransferases that attach carbohydrate to the membrane protein by forming O and N glycosidic bonds. Because of a phenomena called contact inhibition, normal cells usually stop growing when their surface touch. Cancerous cells with the altered cell surface do not recognize the message to stop growing, so they pile up on each other. Here, I told you that there is glycoside transferases which are required, but if there is the problem in the glycoside transferases, इन glycoside transferases का काम ये होता है कि they attach the carbohydrates to the membrane protein. कार्बोहाइड्रेट्स को मेम्ब्रेन प्रोटीन के साथ ही अटैच करवाते हैं और इनमें ओ एंड एन ग्लाइकोसिडिक बॉन्ड्स मौजूद होते हैं। जब इनके साथ नॉर्मल फिनोमेना में ये होता है कि नॉर्मल सेल्स जब ग्रो करते हैं इनके साथ आकर इनका कॉन्टैक्ट होता है तो ग्रो करना बंद कर देते हैं। बट कैंसर सेल्स में बिकॉज दीज आर नॉट प्रेजेंट, ये वहाँ पे नहीं होते, इस वजह से उनको सिग्नल नहीं मिलता कि यहाँ पे ग्रोथ रोकनी है। कैंसर सेल्स फिर उनकी जो ग्रोथ है वो इंक्रीज होती रहती है। अल्टीमेटली होता है क्या है कि वो इस सेल्स के ऊपर ही एक दूसरे को स्टैक करना शुरू कर देते हैं। प्रोटीन्स टर्नओवर ओलिगोसैकेराइड्स आर आल्सो यूज्ड टू mark proteins for the age. Glycoprotein in the serums are constantly turnover. Glycoproteins in the serum are constantly turned over, the newly synthesized ones, taking the place of the aged ones. The proteins exchange होती रहती हैं, मीन्स चेंज होती रहती हैं, स्पेसिफिक एज है इनकी, उसके बाद इन्हें टर्न ओवर होना होता है। In general, removing of the carbohydrate units from glycoproteins almost always increases their susceptibility to proteolytic degradation. Turnover of the serum protein is initiated by the removal of carbohydrate residues by the hydrolysis. Say, for example, this is the protein that contains the serum, it contains the sugar group. This is the sugar group attached with this protein. Upon the addition of, upon its hydrolysis, this residue, these sugar residues will be removed. When the sugar residue will be removed, the degradation of this protein or turnover of this protein will be started. Liver cells have on their surface receptor protein sites that recognize aged glycoproteins lacking sialic acids. Such glycoproteins are taken up by the liver cells and degraded by the proteolytic enzymes to free amino acids. Viral growth. Many viruses, including herpes simplex, hepatitis B, influenza, and HIV virus, these viruses contain a protein on their surface that have potential site for linkages of oligosaccharides. Carbohydrates may be linked to these proteins using host cell enzymes to produce glycoproteins that are indistinguishable from those on the host cell. This allows the virus to thrive because it avoids immune surveillance by the host cells and can also attach and fuse to the host cell receptors. Virus contains virus contain uh, the proteins on their surface that have potential sites for the linkages of the oligosaccharides. Uh, these proteins can be attached with the oligosaccharides. Carbohydrates may be linked to these proteins. The proteins of the virus can be attached with the proteins. And virus tend to produce the glycoproteins. The virus use the machinery of the host cell. Or we can say the carbohydrate may be linked to these proteins using the host cell enzymes to produce glycoproteins that are indistinguishable from those on the host cell. So the virus use the host cell enzymes and produce the and produce the glycoproteins uh, or use the host cell enzymes and attach the carbohydrate with that of the proteins of the virus. So the viral proteins cannot be distinguished from the host proteins. This allows the virus to thrive because it avoids the immune and surveillance by the host cell and can also attach and fuse to the host cell receptors. So because of these glycoproteins, uh, immune response cannot be generated. For example, HIV enveloped protein one contains the 120 has 20 to 5 asparagine residues, 120 amino acid, 120. Uh, for example, the HIV enveloped proteins, uh, 120 GP120 has 20 to 25 asparagine residues that are potential sites for attaching of the oligosaccharides using oligosaccharides by the N-glycosidic linkage. So we can take the example of the HIV protein GP4 by 120. It contains 20 to 25 asparagine residues. And we people know here that asparagine is the site. Here you can see the asparagine is the site. Asparagine is the site where the asparagine is the site where the other monosaccharide units can be attached. Antifreeze glycoproteins, fish living in the sub-zero waters of the Antarctic and Arctic are protected from the freezing by the presence of anti-freezing glycoproteins in their blood. The glycoprotein act by lowering the freezing temperature of the water. Each of these protein has a large number of repeating units consisting of tripeptide, alanine, threonine, and alanine, threonine, and then again the alanine, and a disaccharide, usually B-D-galactosyl 1,3 alpha N-acetyl, or we can say that it may contain the beta D-galactosyl, and it is linked to the 1,3 with the alpha N-acetyl galactosamine, and they have the covalent linkage with the threonine hydroxyl group, and they make the O-glycosidic bond with the threonine. Japanese biochemists have found that the proteins having only a single tripeptide unit with disaccharide causes a measurable lowering of the freezing temperature of water, although the naturally occurring glycoproteins usually have several tripeptide disaccharide units. They have shown by NMR spectroscopy that the glycoproteins fold to expose a polar hydrophilic site that interacts strongly with the ice crystal, thus lowering the freezing temperatures. It will be all about it is the all about the carbohydrates. I think it is enough for you all. It is enough. For further, we will discuss about the lipids or proteins. Up till then, thanks and Allah Hafiz.